next up, we've got Dr. Steve Millar. Steve's worked in Canada at Guelph and has recently come to New Zealand about 18 months ago. Yep. To lead the team at Invimay AgResearch. So I'll hand over to Steve. Thank you.
Okay. Thanks, Graham. It has been about 18 months, and I think the first time I met a lot of people in the beef breeding game was at this same event last year here in Fielding. And we were looking at that time to get semen on bulls that were kind of unique in Hereford and Angus breeds, and there's breeders in the audience here that put semen up for that project, and I'll talk a little bit about it today. Okay, so we're going to talk a little bit about genomics and sequencing, and the best analogy I've heard about just what does it mean to sequence an animal, you know, how big is the genome, came from a fellow by the name of Jim Womack, and Jim Womack was at Texas A&M, and he's involved with sequencing the first bovine, which was a Hereford, and basically what he was describing, it's nice to look out at the audience because I think everyone here probably knows what a typewriter is. I had a problem in teaching undergraduate students that they were using an old computer program and the student was frozen looking at the keyboard, and I said, what's the matter? And he said, it says hit return to continue. And I said, yeah, hit return. He goes, none of these keys say return. And I didn't realize it actually says enter now. It doesn't say return anymore, but you'll know what a return is. A return is a carriage return, right, on a typewriter. So Jim Womack said, imagine if we had the DNA sequence on an animal, which is made up of basically 4 characters. We have a typewriter that just has 4 keys on it, a G, a C, a T, and an A, and we're going to basically type out the animal's DNA code. And imagine that we're going to type on this 4-letter keyboard, and we're just going to let a ribbon of tape come off that typewriter, and we're not going to hit return. How long would that ribbon of tape be? So this is a ribbon of tape that is typed out in, 10 characters to the inch or 12-point font that you might see on the piece of paper in front of you. And so we could just say this cow here, we might start typing out her sequence, and we'd start typing, and we get this ribbon of tape, and it would basically run from KAITAIA to BLUFF and back twice. So if you can kind of imagine that ribbon of tape running up the road here somewhere through New Zealand in those little letters that you could read, that's a lot of information. So it's basically a huge undertaking to sequence these animals, and it's a lot of information. So why do we do that? Well, if we were to line up the sequence on 2 animals, like line up the tape on 2 different animals, we'd see there's differences. So we'd expect differences because people are going to go out there today and buy a bull and pay more for one than they will for the other because there's genetic differences between them. And what causes those differences, and the differences are caused by differences in the DNA. So here we've got a difference. The one DNA sequence has got a G at that one location and the other has a C. Other than that, they're the same. So you might think, well, geez, you got letters running from KAITAIA to BLUFF and back twice. You switch one letter, what difference is it going to make? Well, in some cases, it's not a difference we could actually identify. We don't really know what. Nothing we can measure. Other times, it's a huge difference. It might be the difference between a live calf and a dead calf. So that's some of these things are really important, and some of them we're still trying to figure out how they're important. They probably are important in some ways. So how many of these potential differences are there? Well, if we just followed the tape along from KAITAIA to BLUFF and back, there's basically one of these differences exists about every foot. So there's actually 30 million differences that we've identified in the Bos Taurus type breeds. So in our Angus, Simmental, Hereford type breeds. Once we throw in the Bos Indicus, the Brahmins and things, it's a whole other ball game. There's a bunch of more differences there, but we won't worry about that. Thankfully, you can be thankful for one thing in New Zealand, that is you don't have to worry too much about a big Brahman cattle industry because it adds a whole layer of complexity for animal genetics. So all this talk about genetics and genomic predictions, essentially it's as simple as we've got these differences and we want to relate them to traits. And the focus of our beef and lamb genetics project is really traits of commercial interest to breeders. And Graham and Jason have already identified that's fertility, condition score, weight traits in commercial conditions in New Zealand. That's what we're trying to do. But when we genotype that animal with, say, the HD50K in the case of Angus and what Kent Anderson talked about, we don't actually genotype every one of those SNPs that exist every foot along the way. We actually genotype about every 150 meters from KAITAIA to BLUFF and back. So we're kind of skipping over large chunks of the DNA. And you might say, well, there could be some really important stuff. You know, you just jumped over, you leapfrogged over a whole bunch of differences. You didn't even look at them. But as Kent showed, actually the tool works really well. He's got 10 to 15 progeny equivalents with his DNA test. That's a pretty accurate test. So how does he do so well? Well, the reason he does so well is that we actually, when we inherit DNA from our parents, we actually inherit it in big chunks. So there's big pieces of DNA that get inherited from parent to offspring, and we can track those chunks with those markers. So I use the analogy a little bit, you know, where we're up to with the technology. I don't know, in the South Island of New Zealand, I notice, you know, duck shooting is a big thing. And I don't know, do people shoot ducks on the North Island? Is that, yeah? I was kind of anticipating, that was a bit of a loaded question. And when you're out there shooting ducks, you use a shotgun, right? And shotguns work great for ducks at, you know, 30 yards or 40 yards. But, you know, a rabbit out at 150 yards, a shotgun doesn't work very well. So essentially with genomic selection, 50K test, you know, we're kind of have a nice shot pattern scattering up the genome. It works well, you know, where we've got 15 progeny equivalents in Angus. That is definitely working. It's working really well in Holsteins. But what's the future? We think the future is drilling down to, you know, those SNPs that we've skipped over. Some of them are really important. So, you know, what the sequencing now is, are we going to put the shotgun aside and get out the rifle and essentially focus in on individual SNPs? That's why we're doing the sequencing, because we think in the long term that's going to add accuracy. So what about genomics? So the first thing is, there was some talk earlier about, you know, how big, I think it was Joe asked the question, how big of a population do we have to genotype in New Zealand? So a lot of what we're going to do in New Zealand is going to be in conjunction with Australia, because you got a joint genetic evaluation system, and you've actually got quite a bit of DNA that's crossing the Tasman. So in Angus, for example, there was a study done, goes back to 1998, basically looking at what's the genetic relationship between performance in New Zealand and performance in Australia. And basically, the genetic correlation for most of those traits is like 0.99. So there's huge similar genetic relationships between New Zealand and Australia. So the good thing is we don't have to worry necessarily about just New Zealand's data versus just Australia's data. We can actually likely put a lot of that data together. So we're going to collaborate. I think that's quite important. The other thing we're doing in the project is we're collecting phenotypes and DNA here in New Zealand. And, you know, and why is that important? Well, if we want to validate the tool, so if we're going to do a joint evaluation with Australia in terms of genomics, breeders want to see in their animals, you know, how does that genomic test work? You got to build confidence, essentially. You know, and in America and Canada, the different states and provinces, they all have their own number plates, and each number plate has got a little tagline. So where I was from in Ontario, it might say, I got a red card, so that's 2 or 5 minutes. It might say, yours to discover, for example. If you're from Missouri, it says we're the show me state. So when someone says, you know, show me, you say, I'm from Missouri. So I think people in New Zealand, they're like you can say, I'm from Missouri, which means show me. So if we want to show you, we have to actually generate our own phenotypes and genotypes. So as I mentioned, we've joined the 1,000 bull project. There's actually 1,700 bulls in this project now. So I just talked to a colleague across the Tasman a couple of weeks ago. They're just about to run the run that's going to include the New Zealand genetics. So we put in 29 different bulls across three different projects, including the ones we did here with Angus and Hereford. And that's one of the bulls that went in from the Angus side. So the question is, well, this, you know, okay, you're sequencing lots of bulls. Why? Well, as I said, we want to drill into what are the genetic differences causing the differences that are important to breeders. And the example I have here is fertility. So there's huge Genetic differences for fertility. If I think as a breeder, you might think about, I bred a cow, but she recycled again, but she was kind of off cycle. She didn't come in 21 days, was a week or so later. She did conceive, but that embryo basically aborted in utero. So there's lots of this early embryonic death that's actually due to genetic mutations. And so we can drill down and identify a bunch of those and turn them into better predictions of fertility. We have one component of the project is we're looking at a special population we have in New Zealand, which was this Jersey Limousin cross population, where we actually have a lot of really interesting and important, expensive to measure phenotypes, including meat quality traits. And the question is, well, why are you looking at Jersey Limousin? I've got Hereford, I've got Angus, that's not relevant. But what we're going to do is we're going to identify with that population, what are those causative mutations causing differences in carcass quality, and then we were going to collaborate with others who have Angus, for example, to see how well it predicts across breeds, which is one of our. So what are we doing? So Jason talked about the progeny test. We're on farm also in commercial herds and some stud herds measuring body condition score and fertility outcomes in females. So the cows that Jason mentioned and Matt and Richard mentioned that are being measured in the progeny test farms, those traits that are measured on those cows get combined with the traits we're measuring and what we're calling kind of the tier 2 progeny test. So our goal is 10,000 females here during the life of over this current 5-year program that we have up and going. So we basically have 3 years to go. And it's primarily Angus and Hereford that we're on doing these commercial genotypes. And Carrie Lewis is here from Mount Linton. And just some, so we're very fortunate to get to places like Mount Linton to do these measurements. So just to give you some idea, there's the number of animals that we looked at last year. I remember in the one day, 10,050 heifers through the chute, DNA sample, condition score, weight, across 3 different yards. I learned a new term in New Zealand. They talk about, I'm knackered. We don't use that. I hadn't heard that in Canada before. I know what that means. So getting these takes a lot of work and a lot of dogs, I found out, to push 1,050 heifers through the chute. And this is just Anna Boyd and Trevor Watson from AgResearch chute side there. So a lot of hard work to get a lot of good data. And so not the question, maybe I'll address it. The question was, what are we doing for DNA sampling? So this is Allflex. Basically, Allflex TSUs is what we're taking on all those animals to store DNA for long term. So we could come back to that in questions later. And it's always a beautiful day whenever we're in Southland measuring cows. Okay, thank you.